All right, we're gonna prepping for karaoke already.
All right.
Not gonna break out in song, although, no. Still thinking about a good Nena song or something. Okay, yes, my accent is Austrian, that's where I'm from. I've already introduced myself. My background is, as I mentioned, coming from academic neurology into NINDS, got really excited about clinical trials and clinical trials methodology. In 2009, a while ago, when I participated as a mentee in the predecessor of this course, and this truly sparked the fire in me. Truly, this really informed, the way how I, moved forward with my own career. So I'm really excited to stand here today and talk to you about a topic that somebody should have told me back then in 2009. Probably they did, and I was probably busy preparing my protocol. So I hope you're not busy preparing your protocol right now. Although, preclinical research as a clinician, you may think boring. No, that's important, and it's actually not that boring because it actually opens a lot of doors for you and answers a few questions to the questions that you may have. Coming off of my own CTMC course, our clinical trials methodology course 2009, I was eager to submit my protocol not only to the IRB but also to the FDA, and I did that, and I got slapped with a hold around my head because one of the questions of the FDA was we need additional preclinical data to justify your doses and what you're doing here, so go back to the drawing board. So I was able to redo my protocol, focus on the compound that was okay to move forward, but it was a good learning for me. So I hope you're not going to be in a position where the FDA tells you to stop and go back into creating more preclinical data, because it's probably not what you're going to do, because you're all clinicians and you should do clinical trials, and you need to find other people to do that. Usually it's very expensive, so you want to maximize the information that you can get out of data that may be available to you. So this is what we're going to talk to you about preclinical research. My own journey, as I mentioned, I transitioned to the bright side of industry about 8 years ago, which is also a continuation of my work back from the NIH. I spent some time also at the FDA. There's a wonderful NINDS FDA Clinical Trials Methodology and Regulatory Science Fellowship, if you're considering that. Where you learn lots about also the FDA perspective, which eventually led me to join the company and do for a living what I've been doing before, just design clinical trials. And we already heard before from Chris, we design programs, development programs we call that, which is a series of clinical trials. When you think about your next study already while you execute your study that you have at hand. So what we will talk about today is the role of preclinical research from two perspectives. So the first is the efficacy perspective, and the other is the safety perspective. Because at the end, you have to be a critical consumer, and you have to be critical, and you have to consume that data, because that informs how you move your things forward. We'll talk about elements such as reproducibility of preclinical research. So you need to be critical of what's in front of you. You need to be able to understand that and explain that to yourself, to your funders, also then eventually to your patients when we think about risk-benefit, etc. Talk about rigor and transparency from the efficacy perspective, then we switch over to the safety perspective. That's important because that informs which dose you should give. That's an important piece of information when you start your clinical trial, and also how you set up your safety monitoring procedures in your study, and that's all based on what the compound, what the drug, what the device, etc. is doing, or had been doing prior to you initiating your study, and that may be coming from preclinical data. Actually, having said that, I always say that every year, preclinical is something, and we'll talk about that. That is not something that ends with the beginning of clinical experiments. Preclinical or non-clinical experiments go on all the way through also clinical phases of development. We'll talk about that. So that's an important piece. So we start on the efficacy side, and there's especially in neuroscience, we're all neurologists and neuroscience, neurology and neuroscience is a field that had been historically very tricky and very tough for treatment development. Companies leaving neuroscience several years ago because the success rates in neurology have been so low compared to other fields of developments. Thankfully, in the past years, there's a resurgence of interest in neuroscience. Lots of business happening there, which is good. Lots of investment, lots of new developments coming forward. So you may enter a new wave of neuroscience treatment development. So this is why reproducibility is so important, because we really have to rely on the data that has been generated before we go into the clinic, and we really look at the field that has been struck by what we call the reproducibility crisis. There's data that demonstrates how, that in a survey where over 1,000 scientists said more than 70% could not reproduce in the lab's experiment. Even difficulties not being able to reproduce your own experiment in your own lab. I have certainly experienced that at the NIH itself. Tried to reproduce earlier findings, and not every time this worked. Also certainly in industry, this played a big role, where companies like Amgen looked at data and publications that were out there from preclinical research and were only able to reproduce 11% out of these studies in-house. That really led to a big build-up of industry internal labs, not relying on academic labs and publications, because there was a sentiment out there that academic preclinical research is difficult to reproduce. Irreproducible research is very expensive. Estimates say that about $28 billion per year is spent on preclinical research that is not reproducible. Again, it is important for us to do experiments and to report on experiments. So, it is it's not the case that a dollar that is spent on an experiment that is negative or that does not prove your hypothesis is a bad dollar spent. It's only a bad dollar spent if this experiment is not done well. And if you think about the causes for why often experiments are not reproducible, and we'll talk about that later, because these are now factors that NIH or other funding agency may look at in your experiments. Things like biological reagents and reference materials, things that don't really come to our mind for clinicians what to think about. How can we even interpret whether a cell line is adequate or not? That's the majority of the costs is going into areas such as that would then downstream lead to irreproducible results, just the reagents that go into experiments. So this is why it's important that we know about these things. These things are disclosed, and there's transparency around that. So at least we can go to colleagues that really know about that, who can tell us whether that's an experiment to rely on or not preclinically. There are examples out there. There are multiple ones. Every year I bring this one because that's one of the striking ones also in neurology. If we think about the ALS story around early SOD1 approaches, now there are treatments, thankfully now, although effect sizes are not great, but some treatments are in the pipeline and even across the finish line for SOD1. But early approaches with minocycline, for example, showed several groups producing positive results on survival in SOD1 mice, leading to a randomized controlled trial with over 400 patients treated for 9 months, only then to be shown that minocycline did not have any benefit in patients with ALS. So the question is, did these mice experiments translate to humans? Is it a model problem, or is it an experiment model? And one of the big takeaways, and this is not just the factor for ALS, and we'll talk about others as well, is that we have to be always vigilant about things like effect size and sample size and cohort size and the relation of small sample sizes and inflated effect sizes. What do I mean by that? This is a study that looked at the cohort size of mice per group. So we're talking about really, experiments in mice. How many mice are allocated to each group, and the smaller the sample size of the group, the higher is the effect size. That's interesting, right? So and there's a relation between cohort size, sample size, and an inverse relationship between sample size and effect size. And there is also a relationship here in color-coded are quality attributes in red. You know, even at the small sample size, the effect size goes down once you increase your rigor of your experiment. In that case, these are matched and balanced cohorts. So the higher essentially your quality criterion, if you introduce randomization, if you introduce matching between groups, that somehow has an effect on effect sizes. So that's really, for me, kind of the takeaway is that sample size plays a role, and you want to make sure that you are looking at an actual effect size and not a noise. And a retrospective study looked at the actual statistical power in neuroscience preclinical experiments, and that in neuroscience is really abysmal, 21%. So you really have to be careful when you interpret your preclinical studies. Same thing is true for stroke. This is also a meta-analysis across multiple studies, and they assigned a quality score. The higher the score, the better the quality in a particular study. And also you can see the lowest quality studies had the highest effect sizes. So the more you control for potential biases in a clinical trial design, the more likely it is you get a true effect size, which is probably lower than you initially thought it would be. Also here, when we talk about reporting of preclinical experiments, also meta-analysis here again. It's a little bit dated here, but this gives you some indication about what we often see or don't see in animal trials. Usually, the things that we see the least in animal trials are things like randomization, adjustment for multiplicity, and blinding. So when we talk about these key quality criteria in clinical trials, same thing is true for non-clinical trials. So be vigilant about the trials that report wonderful effect sizes that you want to see, but not talk about things like blinding, randomization, adjusting for multiplicity. Yes, these things are also important in non-clinical experiments. So that essentially led NIH to think about what are we going to do about that? What could be factors that lead to this crisis of reproducibility and lack of reproducibility? And there are many factors: lack of sufficient training in researchers in experimental design. I mean, you guys, are at the top of your class of trialists and clinical trialists. For you, it's probably straightforward how to approach that, but we also have to bring the message to our non-clinical colleagues in the labs, in front of animals. There's, of course, in the career of people, it's better to publish positive studies, of course, right? You have a publication bias here. We may be pushed towards provocative statements. You may be pushed towards yourself to make over-interpretation of your data. How often we have cured Alzheimer's disease in mice, right, over many decades, rather than essentially reporting these as hypothesis-generating experiments if they weren't adequately set up and powered. Often these publications lack information on basic elements of experimental design. If they don't talk about randomization and blindings, probably not. That probably didn't happen. Negative data is often not published, and it's of course difficult to interpret data that is not published. And to access data. So you may be, as you work out your biological rationale for your intervention, and you may reference 3 preclinical studies that have been published. Think about that. This is already probably a subset of a biased sample, and if you go then with a study with the highest effect size, you may be biasing yourself into an overinterpretation of an effect that may not be there. This is a topic that's now, for many years now, being looked at very carefully in NIH grant applications. So your grant will be reviewed based on also the scientific rationale that you bring in front of the agency, and there are certain criteria that you need to reflect upon in your grant as you are relying on preclinical data. So what is the rigor of the prior research? So you need to think about and discuss strengths and weaknesses of the published research or the preliminary data that's in front of you. You have to discuss the experimental design in terms of robustness and unbiased and potential bias of results. You definitely have to consider certain relevant biological variables. I mentioned before that matching and balancing is important for non-clinical studies as well. And you have to have some idea about authentication of key biological and chemical resources. That's often for us clinicians most difficult to do unless you have experience working in a lab, but as shown before, it's one of the key drivers of lack of reproducibility, that the cell lines were biased, or there's certain antibodies or chemicals that were not comparable across studies. So these are all things that you need to be aware of. You need to judge your literature or your data upon when you set up your own experiment and put it in front of the NIH. So the next part of my talk is about basically the question now, as you plan your study, as you plan your submission to the FDA, what questions you have to put in front of your non-clinical data package, or in general, in front of your clinical trial plan. So if you, let's say, plan to administer a drug to humans, the overall question is it safe to put the drug into humans? And if so, what is a safe dose for human clinical trials? And by that, we often are not just considering a single dose here, but we consider things like what is the starting dose, what is an end dose? So what can you start with? What is the highest dose that you can give? What I'm not covering here is something that we discussed in a small group before. Dose is only one element. The next thing that you have to consider is something that we call exposure, right? Because as a certain dose, if you give that to 5 different people, you might give 5 different drug levels within these organisms, for example. So what, this is the pharmacokinetic part of that. So you have to consider that as well, and you have to have some understanding what your drug does in a human body. You definitely have to know your dose-limiting toxicities, what to look out for when you administer the drug in your clinical trial, and which safety monitoring is appropriate based on the properties of your drug and based on the actions of your drug, and also what are potential toxicities that are difficult to monitor in clinical trials, right? So that's something that you have to think through as you plan your protocol. So the general principles for non-clinical testing is to identify organ toxicity, identify the relationship to drug exposure, right? How does a dose relate to exposure? Because exposure is ultimately what has the effect on your target organs. What are on-target effects? What are off-target effects that may drive your safety profile? How relevant are these to humans if you're looking at non-clinical testing? What's the predictive value of non-clinical findings? And what are potential safety biomarkers to monitor for in the clinic? So as long as you know how to monitor for that and intervene if necessary, like if your sodium climbs above X, then you pause, or then you de-escalate or something like that. So it is all about your tools that you have in hand to keep your patients safe, but at the same time conduct your experiment in the way you've planned it as efficiently as possible. So the non-clinical safety testing regimen is depending on multiple things. So there's now the one box fits all, and that's depending on the type of therapeutic you are applying. We're talking about small molecule and biologics. We're not talking about devices today, but this is really around drugs. What is your therapeutic indication? Is this a CNS targeted intervention, or is it periphery you target? And also, what is the scope and design of your first human trial, right? So it's always in relation to that specific trial that you are planning to do. That will shape your non-clinical package. Why is that important? Because if you're studying a drug that is an investigational drug, you have to file an investigational new drug application with the FDA. If it's not an approved drug in that indication, we'll talk about that. And this IND application, if you have ever seen that form, you have to actually sign that. It's a 1571 form, and that actually has some legal ramifications if you read that. So there's actually some laws behind there. So you really want to understand what you're signing here and what you are committing to submitting to the FDA and committing to knowing yourself what you're getting yourselves into. So this 1571 form is basically, this is a table of contents of things that you have to submit with your application that includes a few forms, and most important, your protocol, right? That's your study protocol. But at the same time, also there are things like chemistry, manufacturing, control data, pharmacology and toxicology data, previous human experience. So these are 6, 7, 8, and 9. These are things, data pieces that we'll be talking about today that really shape your thinking and your protocol as you go forward. So what should you know about your drug? What does chemistry, manufacturing, control stand for, CMC? What you have to know, a drug is not just the active substance. It contains a few things. It's the drug substance, the API, the active pharmaceutical ingredient. Then there are additional things in it, like excipients, that actually make your drug into a tablet, make it stick together and not dissolve. There are impurities, and there's, depending on where you are in your development, there's some levels of impurities that are accepted or not, and there's the container as well. That is also part of your drug, how you put it in front of patients. And then there's data on the identity, strength, purity, and quality of the drug. So these are all analytical approaches how to describe a drug. Then there is additional information: how do you manufacture it, how do you store it, stability, things like can you leave it on the table for X hours before you administer it? How much time do you have between thawing it and administering it? That really informs how you have to handle your drug in the clinic. What about pharmacology and toxicology? So you want to understand the pharmacological effect and mechanism in animals. If your drug has never been given to humans before, that includes things like absorption, distribution, metabolism, excretion. ADME is the abbreviation for that. So we'll talk about toxicology. And there are some safety pharmacology studies that may be required for certain target organs, depending on what you're studying, depending on the property of the drug. There may be some special cardiovascular studies needed, CNS, pulmonary, etc. There may be specific toxicology tests that may be required, depending on your mode of administration, for example, dermal toxicology, etc. Genetic toxicology is also, these are often in vitro testing for long-term administration studies and later stages of development needed. Once you're in your first in-human study, are you done with preclinical? The answer is no. We talked about that before. You go from your drug discovery phase to your preclinical phase, and then you finally go into clinical trials. And as I mentioned, for your first in-human study, you may be fine with your chemistry, manufacturing, control just for your phase 1 product. It doesn't yet need to be commercial grade, nicely packaged drug pack as you use in phase 3 or later studies. It can be adequate just for phase 1, manufactured just for the purpose of a small single-site study. Your pharmacology package can be as simple as supporting your first human study, which is never more than 7 days of something like that. Acute toxicology maybe sufficient for that. But once you go into later stage of development, you give your drug 3 months or 6 months, etc. You need additional preclinical data for that. Chronic toxicology studies for 6 months, for example. Pharmacology of alternate formulation, if you switch from an oral to a subcutaneous drug or the other way around. And other non-clinical stuff. So preclinical does not end with entering the clinic, so that's the takeaway piece here. So non-clinical development, it goes all the way past approval sometimes when you introduce new formulations, for example. Okay, so this is another busy slide here, but essentially the takeaway is that for any data you cite, and that is in your application, make sure you know the specifics of that data. So you want to know, how many were exposed, what doses were given, what was the duration of the administration and the route of administration. And something that's called the investigator's brochure. It's something that's often not required if you do a sponsor investigator single-site study, but once you get into a multi-site trial, or you are administering your drug that's under investigation by another sponsor, such as a company, for example, then they have to have an investigator's brochure. This is a confidential document. You actually have to sign a confidentiality agreement before you can access that. So you're not allowed to talk to anybody about that, because these are often company secrets, not published often. And that's regulated by the FDA, for example, and not just by the FDA, but sort of the global ICH guideline around what an investigator's brochure should contain. It's exactly all that data that I'm telling you that you should have visibility to. So you can read there what toxicology studies have been done, which pharmacology studies have been done, which animal models have been done, what's the CMC properties of your drug. So make sure you read that through and you really understand that. That's something that an investigator actually needs to sign off, make sure that you understand that. And this will then really drive, answer some of your questions. If another sponsor has an IND open, they may not be providing you all of these study reports, but you can obtain something that's called the right of reference letter. So the FDA then actually could look into that other sponsor's package of all the studies that they have done, because once you submit an IND with a complete new drug, you have to submit study reports of all these preclinical data. So the FDA at least can take a look at that. If you have access to study reports and data sets, get those. Ask the sponsor, do you have actually the study report of that particular toxicology study, if you want to understand a little bit more what happened in the study. And there's often more information in study reports than in publications, never rely on abstracts. So it's even prior human studies, no AEs were reported. If there were no AEs reported, then you have to be worried about that, because they may have happened, but they're not reported. So there are things like that you want to understand. So the goal is really that your planned dose, duration, and route of administration should be supported by what's been done before. So there are a couple of scenarios here. What if you're dealing with an approved drug and you use it per label? So per label means there's often a dose that's been spelled out in the label as an approved dose, a route of administration, and the indication may not be the same, but maybe closely related to your indication in terms of overall risk-benefit. Then you may be okay with the label as such, and you can submit and open your IND, so that once a drug is approved, that label essentially replaces the investigator's brochure. Once you change anything that's spelled out in the label, you give a different dose, give a different route, different patient population that has impact on risk-benefit, there could be additional non-clinical asks, or you have to make a case for why no additional non-clinical data you want to provide. You always have the opportunity to have a pre-IND meeting with the FDA, where you can actually ask these questions. Do I need another, or is this sufficient, what you put in front of them? If you use it as supplements, often a question, nutritional supplement or other supplement. If you use it as a drug, it is a drug, and it's considered to be a drug, and then all the standards still apply. If you manufacture a drug yourself, then you have to expect that you have to submit a full non-clinical package on your material. What about picking the right dose now? Let's say, okay, we are through our toxicology study, we have all our data available, we know which doses had a pharmacological effect in my non-clinical study, and we know which doses led to toxicology findings. Then we are at the point where we can pick a starting dose. That's usually very exciting, where we integrate all the data that's in front of us. FDA has a lot of guidances available to you. So everything that we talked about before about IND submission suggests you go on the FDA website, pull the IND guidance. It says guidance for industry, but it's also relevant to you. It's basically very nice documents that explain it in plain English, how you should handle that. Okay, so there's a guidance around picking the right starting dose based on the non-clinical data that you have in front of you. So we'll talk about these 5 steps that actually spells it out. These are the 5 steps that you need to do to come to your starting dose in your first in human study. So we're really talking about first time in human administration. First step is the NOAL. This is a really important abbreviation. that we use in our field is the no observed adverse effect level. What is that? That's the highest dose level, usually in animal experiments, that does not produce a significant increase in adverse effects in comparison to the control group. So, so it's basically the highest dose level that did not produce any relevant adverse effects in that animal species. And we're counting adverse events that are biologically significant. We're counting these adverse events for the determination of the NOAL. So that really depends on how many doses were tested. If you did not see any toxicities in your tox study, then your highest dose, by definition, is your NOAL. And often that can be an issue, right? Because maybe you didn't explore high enough. So the goal of these toxicity studies is actually to get an idea about what is the tox signal in this species, because that's what you can then monitor for. So it's a benchmark for safety, and it has to be derived from appropriate animal studies. We talk about which animal studies may be serving as appropriate studies, and they serve as a reasonable starting point to determine where to start for new therapeutics. Next step is that from that NOAL, we calculate the human equivalence dose. So we use the multiple ways of doing that. A very simple but still acceptable way of doing it is scaling by body surface area. So we can convert a dose, usually milligram per kilogram, using standardized species-specific scaling factors. And this comes right from the guidance, and then you can look up your animal species that you may have tox data on: mouse, rats, dog, primate, etc. And then compare to humans, and then there's some conversion factors that you can use. So you apply conversion factor to convert the animal to the human dose. Next step is species selection. What if you tested more than one species? Typically, FDA requires 2 species for a novel, for a new molecular entity before you go into humans. Which one do you pick? So you want to consider which animal model is most predictive of human toxicity. Are there any differences in the metabolic parameters between humans and that particular species? And for biologics, does that model express relevant receptors and epitopes? That's actually really relevant, and it's a new development now. We're talking about that at the very end, where even one can consider no animal experiment may be adequate for that particular drug. So the absence on data of species relevance, you just go with the lowest human equivalent dose. So to basically default to the most conservative way. This is the next conservative factor here, just to be really on the safe side when it comes to your starting dose, and that's a safety factor. And that provides a safety margin, as we say. Because NOAL is the highest dose, no adverse event was seen, so you have to typically, in a dose escalation design, in first in humans, start lower. So where do you start? And that is determined by, that is driven by that, what we call safety margin. And that allows for variability by extrapolating, etc. And the default safety factor is 10. So basically you divide your dose by 10. That's a very pragmatic way, and you can never go wrong with that. But there are reasons for increasing or decreasing that safety factor. You increase your factor if you have a very steep dose response curve, if you have toxicities that are really difficult to monitor for, if you have toxicities without premonitory signs, which is difficult to stop. But there also may be reasons for decreasing the safety factor if your therapeutic is in a class that's very well understood and easily monitorable toxicity. So you have to think about that as well. Last step is you have to have some idea where is your pharmacologically active dose. So there's a criticism around these step 1 to 4 that is really driven by toxicology, that does not take into account at all what your drug actually is doing pharmacologically. So what is the actual pharmacologically active dose in your animal model of epilepsy or whatever, and see how that compares to your safety dose that came out of step 1 to 4. So the PID is the lowest dose tested in animals with the intended pharmacological activity, and you derived it from appropriate pharmacodynamic models, which are the efficacy models in animals. And then you compare your maximum recommended starting dose to your pharmacologically active dose, and if your pharmacologically active dose is lower than your dose that you determined before, up until step 4, you actually select the lower dose here to start. So you don't want to miss kind of the lower boundary of your therapeutic window here. Quick example here, we did 3 non-clinical studies, and we determined the NOAEL is 15 milligrams per kilogram in dogs and 50 milligrams per kilogram in rats and monkeys. So what are we going to do? We convert it to the human equivalent dose, and we use the division method, which we pick our conversion for a division factor here by this column. We divide it by 6.2 for the rat, we divide it by 1.8 for the dog, and we divide it by 3.1 in the monkey. So we end up with 8 milligram per kilogram for the dog and the rat, 16 for the monkey. So we, say, okay, what is the relevant species? You can say both dog and monkey are probably more predictive of humans than rats. In that case, 8 and 16. We default to the lower one, that's 8. So the human equivalence dose is 8 milligram per kilogram. We apply a safety factor of 10. The maximum recommended starting dose is 0.8 milligram per kilogram. Pretty straightforward, right? And then depending on what you're testing here, you compare it to your dose that was the lowest one that is pharmacologically active in your animal model, and here we go. So it's actually pretty straightforward. If this is, for example, for a small molecule, that could be the adequate way of doing it. Limitations, that can be too mechanical. Sometimes it's very toxicity focused, less pharmacology based, does not address dose escalation. That's just the starting dose. So what about the top dose? How high can you go? Often you can go all the way up to your NOAEL, sometimes even beyond that, depending on what the agency is letting you do. There are other approaches, such as the MABEL, also some European regulatory agencies are much more interested in the biological effect levels of your drug. So there's a different approach that uses biomarkers, pharmacodynamic measures, etc. into that calculation. Lots of literature on that. It's the minimum anticipated biological effect level, somewhat related to the pharmacological active dose, it's the MABEL system, and that may be a different puts you into a different dosing spectrum for that. So lots for you to read up if you're in that situation, but just to throw these things out. Let's talk about predicting human toxicities, and that's about how good are these animal models. These are not really animals, these are usually quote-unquote healthy animals that toxicology studies are performed in to predict human toxicities. And so it is not just for your dose selection, but also it's about identifying potential safety signals that you want to monitor for in the clinic. And there are certain species that are more predictive than others. And there are certain concepts or certain adverse event types that are more seen in animals, and some of them are more seen in humans. So for example, if you look at this graph here, things like sweating, dry mouth, headache, dizziness, nausea, you see that mainly in humans. You often don't see that in animals. On the other side, ataxia, convulsions, cardiac sedation, hepatic toxicity, you see that often more in animals than in humans. So you have to be very careful what happens when you see a seizure in an animal. How predictive is that for humans? Often you don't know, but there's a chance that you would not see that in humans. But you have to be aware of that. You have to monitor for that, and you have to know what's going on. Dogs and primates are usually very well predictive for humans, less so mice, for example. So tox studies are usually done in primates, dogs, rats, for example. Then you have to be vigilant about the unknown. In a review, it's about the positive concordance, the sensitivity between observed animal and human toxicity is about 70%. That tells you that 30% of human toxicities are not predicted. There are some examples like, and this it's a separate topic where you have to think about, biologic treatments this is a gene therapy case, for example, the Levine story, Sarepta, Duchenne muscular dystrophy, where we had 2 boys die of acute liver failure with a drug that was approved at that point within 2 months of administration in 2025. And then it was determined that maybe the status of these boys being non-ambulatory played to the risk of this liver toxicity. These are all toxicity findings that have not been seen in the non-clinical studies in that case. So that led to all FDA investigation, and another death occurred a little later, and now this is actually something that is now being investigated by the company with additional immune suppression, because FDA said no, you cannot administer this drug to patients that are non-ambulatory at that point, because that seemed to be basically a flag here for this patient population. But that also tells you that even if a drug is approved, you have to be vigilant that, as we heard this morning, that you will never be able to truly say that your drug is safe until probably you have decades of a post-marketing experience, and the animal models missed lethal hepatotoxicity, and that only surfaced in humans, and even after accelerated approval. So this was an accelerated approval pathway, and it didn't pop out until at the later point. So now the field is learning a lot about the translatability of certain findings in the context of certain therapies across species. And you have to plan for safety monitoring that, you have to potentially plan for the worst case. So you have to think about, serial LFT measuring, keeping patients proximal to acute care, etc. So it does not, this tells you this is not ending this treatment, it just shapes the way this treatment is being administered. First, in terms of patient selection, you reduce your patient population to the ones that you think are not at risk or not at greater risk, and then you can expand probably later once you put adequate safety measures in place, in that case immune suppression, and also handling and safety monitoring to intervene as early as possible as you see signs appear. Last point that I want to make is the FDA is now really challenging the field to move beyond animal testing, at least for certain types of therapies. So it's the principles of the 3 R's: replacement, reduction, and refinement of animal use in preclinical models, and especially when it comes to biologics, there's an understanding that especially our classic non-clinical species are not that predictive for humans, especially if there are some epitopes that are not present. So that's introduced the concept of new approach methodologies, which include several approaches, computational approaches. There is now a whole, effort going on to have, organ chips, for example, and other methods, iPSC-derived models be available to industry and to other sponsors to test your compound before you go into humans. And these organ chips and these approaches are often more reflective of humans, because they are often iPSC cell-derived, for example, from human sources, often better predictive in terms of potential safety signals than animals. So watch this space. FDA's pressure, pushing us, the field, away from animal toxicology studies to hopefully better predictive models. So we'll see probably a change there. It's also going to be much cheaper. If FDA asks you to do a 2-species non-clinical tox study, you think about, we're talking about 7 figures per study. And usually these are not studies that have that high of a scientific value, so it's often difficult to publish on that. But there are grants available that actually help you through these bottlenecks should that be required from you. With that, I want to come to a conclusion. So it is important to think about non-clinical studies. So they are critical to inform your clinical trial, give you often, an answer to questions that you may be, trying to figure out. What is my dose? How should I do safety monitoring? Don't just copy paste from a different protocol. Just really think about what is the risk of your drug. What does your drug do in an organism, and how can you plan your study around that? So you should be able to critically assess the animal studies that have been performed. You should ask for non-clinical data. Go search them out. Look in the IB, see what's published. Ask for the data sets and talk to the regulators. And in the end, human data may be more valuable than non-clinical data. So once you have human data, also from other studies, try to get access to that. Non-clinical experiments are expensive, time-consuming, and if your company is FDA approved and used within the confines of the label, potentially you don't have to worry about that because no additional data may be necessary. With that, I leave you with a few nice views from San Diego. This is UCSD, where I am allowed to teach a little bit. And this is where I work, so if anybody's coming to meetings in San Diego, make sure you knock on our door. We have a beautiful new campus, I'd like to show you around, and then we go to the beach tonight. All right.
Thank you, Dietrich. Nobody bothered to say that you ran all the way from Los Angeles to Las Vegas relatively recently?
You know, I turned 50. I had to do something small. Other than buying a fast car or getting a girlfriend. So.
Probably recommended to run from Los Angeles to Las Vegas. Anybody have any specific questions before we